Hello and welcome to this Bioprocess International Ask the Expert webcast. I'm your host, Alex Nicolaitis, the webcast coordinator for Bioprocess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speaker in the question and answer window on your screen. After the presentation, we'll begin the Q&A portion and I'll ask our speaker your questions. Your questions in the Q&A window will only be visible to myself and our speaker. So thank you for joining us today. It's now my pleasure to introduce our speaker, Derrick Katayama, Lead Scientist, Formulation Development from Samsungbiologics.
Well, thank you very much for the introduction. And it is a pleasure for me to join today's webinar with you and share about Samsung's biologic expansion into the late discovery and early development stage. So just a brief introduction. I'll give a little bit of background on the Samsung Biologics CDO service. So traditionally, the CDO was solely focused on the right side of the slide here, with a focus on cell line, process, analytical, and formulation development. However, realizing that many of our clients would benefit from a CDO service that supports late discovery, Samsung Biologics is expanding its scope to support activities such as early material generation and developability assessment. So what is the value of investing into late discovery? Well, as we all know that there's a high rate of failure observed in development for biopharmaceuticals. Sometimes this is due to gaps in the design of the molecule or selection of the right molecule, and this can lead to issues that are realized later on in development. So in general, some important CMC factors that need to be considered are things like manufacturability and safety. Therefore, using Samsung Biologics platforms S-CHOsient and DEVELOPICK helps to provide an early assessment tool to help fill in those gaps for molecular selection, evaluating attributes such as cell productivity, stability risk during processing, and physical chemical stability and solubility. One critical part about being able to perform a developability assessment is having available material. So often later stages of discovery, there might be multiple candidates with limited amounts of material. In order to perform a developability assessment, SBL offers this S-CHOsient platform. The S-CHOsient platform has been shown to be able to produce transient expressed material from different scales, from small to large. And as this slide here shows, it can go from a small scale to a larger scale and produce material at comparable productivity and product quality. As you can see from here, we see the productivity in terms of titer, the purity profiles by size exclusion HPLC and CESDS are comparable between the small and the large scales. The S-CHOsient system has been shown to be very effective across different modalities, such as monoclonal antibodies, SC fusions, and bispecifics. This system also shows higher productivity compared to a commercial platform, as you can see on the right-hand panel there with the direct comparison to the commercial platform. Also comparing the S-CHOice platform to a commercial platform, we didn't really observe any discernible differences in the product quality and attributes like the size exclusion, CESDS, results, as well as the biophysical properties such as TM and T aggregation. So to summarize this brief introduction of the S-CHOsient system is capable of providing sufficient material for developability assessments for multiple molecules at the stage of late discovery or early development. And another important aspect that comes out of the S-CHOsient platform is it can provide an advanced look at the productivity for that particular molecule. Okay, so now we'll kind of get into the bulk of the presentation, which is regarding DEVELOPICK. And this was the process of actually doing a developability assessment for these early candidates to help select the right molecule. So when we think about attributes that are important for developability, we think of aspects such as molecular stability, solubility, and process stability, like low pH stability for viral inactivation studies. The goal of DEVELOPICK is to help our clients select the molecule that is with the best attributes that will help move forward successfully into development. But let's take a step back for a moment and consider all of the various degradation pathways that a protein therapeutic or a biopharmaceutical product might undergo, processes such as unfolding, chemical modification, fragmentation, particle formation, and aggregation. And these can occur at under different conditions, such as different temperatures, different other conditions like shaking, light exposure, and exposure to different impurities. So therefore, the sooner that we can learn about some of these molecular liabilities, the team is better able to select an appropriate molecule to move forward into development. So now at the stage where we perform DEVELOPICK, it's at the late discovery, early development phase. And that's kind of shown here in the box that's highlighted. And it is at this stage that we help our clients better understand the molecule and their candidates that they are trying to move forward. Some advantages of the DEVELOPICK platform is that the analyses that are performed are short-term, and they use relatively small amounts of material, and we utilize automation to help us perform a lot of these studies. Therefore, at this stage for the DEVELOPICK, we're able to evaluate multiple candidates side by side. So the typical workflow for DEVELOPICK is as follows. We receive the material. We will usually assess the primary structure. Now, this is to help us look at various sequence liabilities for the molecule. We'll talk a little bit about that later. We'll perform a basic characterization of the molecule. We'll expose it to some thermal stress as well as pH stress. And we'll also evaluate the relative solubility of the molecule. And that's kind of the general high-level workflow for developability. So the primary sequence, one of the first items that we do look at is the primary sequence. So, although that the assessment that we do can't really give us information on secondary or higher order structure, but it is a means for us to be able to assess the sequence liabilities. And for example, if we have a molecule like a monoclonal antibody, we take a very sensitive portion like the CDR, and we look for various hotspots. And if it has some of these hotspots like an asparagine glycine, an aspartic acid glycine, methionine, etc., we can assess if one or more of those candidates has less of these hotspots within the CDR, for example. If not, if we're not able to differentiate that, it still lends information to the specific types of solution conditions that we might evaluate to minimize these pathways of degradation. We also will perform a biophysical characterization or a basic characterization study of the molecule. And these are performed with high throughput methods. And it allows us to look at attributes such as thermal unfolding, the temperature of aggregation, as well as hydrodynamic properties, such as the diameter and polydispersity of the molecule. We also have some optional biophysical characterizations that can be performed. In the typical workflow, we don't always implement these, but these are options that can be evaluated during developability. These are things like looking at the colloidal stability of the molecule. So the propensity for these molecules to self-associate, for example, we might assess by using the diffusion interaction parameter or the second virial coefficient. We can also get a feel for the thermodynamic stability by measuring the free energy of unfolding by doing a chemical denaturation using guanidine or urea. And we can calculate what that free energy of unfolding is for that particular molecule and compare molecules side by side. An example of one of the optional biophysical characterizations is one of the molecules we looked at for different solution conditions for a monoclonal antibody. And we were measuring the diffusion interaction parameter. And as you can see, sort of just at a high level summary here, that the more repulsive or the more positive KD that we saw, we were seeing more repulsion of the molecules, and that correlated with, as you can see on the right-hand panel there, less aggregation. So more repulsion, less aggregation, and we were able to kind of assess that in different solution conditions using a fairly small amount of material, using our high throughput methods. So one very common aspect that we will evaluate for stressing the molecule is that of thermal stress. And this is a very common stress applied to biopharmaceuticals. And this gives us a preliminary evaluation of the potential routes of degradation, so whether or not it's going to be aggregation, fragmentation, etc. We can gain some of this information during the developability, and this can also inform future strategies for formulation development. And this provides a seamless transfer of information from the late discovery stage into the formulation development stage. Another assessment that we perform during DEVELOPICK is that of relative solubility. So one of the things that occurs is that at the stage of developability where materials are fairly limited, a true evaluation of the solution conditions that favor solubility are usually not available at this time. So one methodology that we leverage to help us understand the relative solubility between different molecules or even different solution conditions is that of a PEG precipitation solubility assay. And as you can see in the graph here, this is an example of a monoclonal antibody evaluated at different solution conditions. And in the top panel, we're monitoring the concentration as a function of PEG concentration. And on the bottom panel, we're also monitoring and seeing it correlate with increasing turbidity with increasing PEG concentration. But this is a good way for us to be able to compare different solution conditions as well as comparing different molecules. So I'd mentioned this several times. One of the important aspects of DEVELOPICK is to be able to compare molecules side by side. And we can send it through the workflow that we described earlier, looking at the primary sequence, the low pH stability, some basic biophysical characterization, and looking at the physical chemical stability as it's exposed to different stresses, thermal and pH stress. So we'll kind of move into discussing some real-world case studies where we were able to apply DEVELOPICK and help identify the more favorable candidate to move forward. So this is a monoclonal antibody case study. In this case, we have 3 different monoclonal antibody candidates. All were CDR sequence variants, and we were able to apply the DEVELOPICK workflow, looking at the basic characterization, thermal stress, and low pH stress, and to try to differentiate between these candidates. So as you can see from this slide, each of these candidates exhibit different biophysical properties, such as temperature unfolding, temperature of aggregation. And in the table, you can see that there are some subtle differences between the unfolding temperatures and as well as the temperature of aggregation. You can also visually see this in the graph, the bottom left, where we can see that candidate 3 is aggregating much sooner than, say, candidate 2 and candidate 1. Another observation that we had was that candidate 3 exhibited a greater amount of light scattering after the temperature ramp as compared to candidate 1 and 2. So with the moving forward into the DEVELOPICK, we also assessed it by thermal and low pH treatment. And so in these panels here, we can see that on the left side, we were assessing the size exclusion HPLC data for low pH hold. And it was important for us to realize that all 3 of these candidates showed very similar stability under the low pH condition, which was important because that means that they are also available for viral inactivation processes later on in the downstream process, later on in development. But when we compare the thermal stress and we look at different thermal stress conditions on the right side, we can see that candidate 1 and 2 show a lot more retention of the percent main peak by size exclusion HPLC as compared to candidate 3. So in other words, candidate 3 showed the greatest amount of loss of monomer by size exclusion HPLC. Just as a note here, we were able to identify candidate 1 as the most favorable candidate, and that one actually proceeded into development, and a formulation was successfully completed for candidate 1. So for case study 2, this is a case study encompassing a complex protein. So these were comparing two different molecules. One was a bispecific antibody compared to a FC fusion, and these were the two candidates for selection. And again, this went through the typical DEVELOPICK platform, and we performed the basic characterization, the thermal stress, and the low pH stress, and were able to identify some different attributes for these molecules. So in this example, these candidates exhibit a fairly large difference in their biophysical properties, where candidate 1 is showing a higher thermal unfolding temperature as well as a temperature of aggregation. And you can see that in the thermogram from the differential scanning calorimetry on the bottom left, as well as the temperature of aggregation result on the right, where we can see that there is a clear difference between candidate 1 and candidate 2 from this basic characterization study. So here we're looking at the thermal and low pH treatment for these molecules. As we can see in the panel on the left, there's not really any significant difference between candidate 1 and candidate 2 with regards to the low pH stress. However, in comparing different thermal conditions for candidate 1 and 2, candidate 2 shows a greater loss of the percent monomer by size exclusion, HPLC, as compared to candidate 1. So this one was a pretty clear-cut case where candidate 1 was more stable than candidate 2. And we were able to move forward and select the appropriate molecule to move forward into development. So just some concluding remarks. In general, we understand that the development of biopharmaceuticals is a very challenging task, and that there are many aspects to biological molecules that need to be considered during the development. So therefore, being able to select the most appropriate molecule enables success at the very start of the project. And so that's the goal for Samsung Biologics, to provide the S-CHOsient platform and the DEVELOPICK platform to help our clients succeed in taking on this big challenge.
Okay, great. And as a reminder to our audience, you can go ahead and type in your questions for Derrick now. So our first question is, so far, what is the experience that Samsung Biologics has with the DEVELOPICK and its customers?
Yeah, the DEVELOPICK program was launched in October of 2022. And since then, 4 programs have employed the DEVELOPICK platform. I anticipate, though, in the future, DEVELOPICK being used much more because there's a lot of attention being given to understanding the properties of the lead candidates at this early stage of development.
All right, and our next question is, you mentioned that there's a seamless transfer of information from developability stage into the process development stage. Can you elaborate a little bit more? Yeah. So I think that's a great question. For programs, for example, where DEVELOPICK and the materials generated for DEVELOPICK at Samsung Biologics, both our upstream and our downstream teams are involved in the material production, thereby providing the upstream and downstream teams with an early assessment and experience with that particular molecule. In addition, as we mentioned in the presentation, that in the DEVELOPICK, we evaluate low pH stability, and this is not only to help inform the formulation team about different liabilities, it also provides valuable information to the downstream team with regards to viral inactivation for like the low pH hold. And then finally, the developability studies that are performed are always performed within the formulation development group. And it's very often that the same subject matter experts that are working, the scientists that are working on the developability, will continue to work on that formulation development for that program. Therefore, any observations, specific degradation pathways, aggregation, fragmentation, for example, will be taken into consideration for the selection of the various compositions as they move forward into the formulation development. All right, and our next question is, would you recommend using your transient expression service to conduct DEVELOPICK, and if so, why?
Yes, certainly. I would recommend using the transient expression system, S-CHOsient, for the DEVELOPICK service. The material that's generated has been shown to have good quality attributes. In addition, early production of the S-CHOsient system can also provide information early on regarding the productivity using the same host cell as with the stable cell line, the S-CHOice.
All right, well, it looks like we're running a bit short on time, so if we don't get to your question, we'll be passing them along to Derrick, who can follow up with you directly. Please go ahead and continue to type in any questions you have, and we'll get those passed along. The last question for the webcast is, the biopharmaceutical field is a very dynamic and competitive field where speed of development is often an important factor. So I might wonder, is it really a good idea to spend more time to conduct the additional developability studies?
Yeah, no, I think that's a fair question. It's good, but I think that performing a developability assessment is even more important in this competitive and fast-paced world of biopharmaceuticals. One, it enables the selection of the correct candidate to proceed into development, and additionally, the information that's gained from the developability assessment helps to minimize potential delays later in development if there are some issues that arise. Further, the developability assessment can be performed in parallel with other activities early in the process, therefore it does not impact the timeline because the material that's used for the developability assessment is generated by the transient expression system, S-CHOsient, and then we will perform the DEVELOPICK service in parallel with some of the other development activities that are going on. So in short, yes, DEVELOPICK requires additional studies, but it helps set the program up for success from the start.
All right, fantastic. Well, thank you so much, Derrick.
Yeah, thank you very much. Thanks for having me.
And thank you to our audience for joining us. The recorded version of this webcast will be available for on-demand viewing on our website, and as a registered attendee, you'll receive a follow-up email providing you with the direct link. We look forward to having you join us at future Bioprocess International Ask the Expert webcasts. Look for those announcements in your inbox. Bye.